Hello again. Welcome to my fourth video session. So today I'll be doing a presentation on fertilization and post-fertilization events. Here are the objectives that you will be able to complete after this presentation. 1. To discuss the unique fertilization in angiosperms. 2. To compare and contrast internal and external fertilization in animals. 3. To explain the challenges and strategies in aquatic animals for their fertilization. 4. To determine the type of cell division during embryogenesis. 5. To differentiate between oviparous, viviparous and ovoviviparous animals. And 6. To describe the changes in the flower after fertilization. To begin with, let me have a quick review on the events in sexual reproduction. So we learned that there are three main events in sexual reproduction: pre-fertilization, fertilization, and post-fertilization. And I've already discussed with the pre-fertilization events, which includes gametogenesis and gamete transfer. After the gamete transfer, it is then followed by fertilization, which is the vital event of sexual reproduction. Let's look at the cartoon here. We find 3 chromosomes in each of the egg and the sperm cell. However, both the eggs and the sperm has only one copy of each chromosome. And because of this, we call the gametes as haploid cells. And the haploid cells, as we know, are denoted by 1n. Now in this case, 1n is 3. During fertilization, there is fusion of the two gametes. More importantly, there occurs the fusion of the egg nucleus with the sperm nucleus. And this results in the formation of a zygote. Now what about the ploidy of a zygote? Will the chromosome copy remain same or will it change? Let's look at the illustration of the zygote. What you can see here is, after fusion, the zygote cell has 3 pairs of chromosomes, or a total of 6 chromosomes. So this shows that zygote has received a set of chromosomes, each from the egg and sperm cell. Now, by definition, if a cell has 2 copies of a chromosome, or if a cell has a pair of homologous chromosomes, it is called as a diploid cell. And hence we can conclude that zygote is a diploid cell. So to put simply, fertilization is the fusion of 2 haploid gametes to form a diploid zygote. Next, let me give you a brief idea about the fertilization in angiosperms. In angiosperms, we observe a unique fertilization known as double fertilization. And this fertilization involves 2 fusion, syngamy and tipple fusion. Now, do you remember the structure of a mature ovule? Well, look at the illustration here. You can see a mature ovule having 7 cells and 8 nucleus in their embryo sac. Once the pollen tube reaches close to the micropylar end, the generative cell in the pollen grain divides and gives rise to 2 male gametes. One of the male gametes fuses with the egg cell or the female gamete cell and forms a diploid zygote. And this is their actual fertilization known as syngamy. The other male gamete fuses with the two polar nuclei in the central cell and produces a triploid primary endosperm cell. And this fusion is therefore called as triple fusion. Now, since there are two fusions taking place in the embryo sac of an angiosperm, this type of fertilization is called as double fertilization. In animals, we can broadly think of two kinds of fertilization: internal fertilization and external fertilization. Internal fertilization is seen in cases of reptiles, birds, and mammals, and external fertilization is seen in cases of most fishes, amphibians, and aquatic invertebrates. Now, these two types of fertilizations are classified based on the site of their fertilization. If the sperms are transferred inside the female body and fertilization occurs inside the body, such fertilization is called as internal fertilization. And the process through which we have the transfer of male gametes or the sperm into the female body is called as copulation. On the other hand, in the external fertilization, sperms and eggs are released in the water and fertilization occurs in the water. Such kind of fertilization is external fertilization. Now, we can imagine the challenges associated with this kind of external fertilization. The sperms and eggs released in the water must get fertilized. Some of the gametes may get eaten away on its way by certain predators, or some of the gametes may get destroyed by the harsh environmental conditions. Nonetheless, these organisms adopt certain strategies through which they can be overcoming the losses, or they can increase their chances of fertilization. They produce huge amounts of gametes at a time, or they can also ensure that the gametes are produced simultaneously by the male and female partner. Well, so we learned that in external fertilization, the success of the fertilization is highly uncertain. Now, what about the case in internal fertilization? The number of eggs produced in organisms with internal fertilization is much lesser. Now, why this is the case? This is because there is greater chance of fertilization since the gametes are fusing inside the female body. And in addition to this, there is more of parental care and safety for their survival. Well, after fertilization, the diploid zygote then proceeds into post-fertilization events, which means to say the zygote grows into a multicellular embryo and eventually into an organism. And this process can be understood through two basic events: cell division and cell differentiation. The type of cell division during embryo development depends on the ploidy of an organism. As shown here, if the organism to be formed from a zygote is a diploid organism, then the zygote would undergo mitosis, since mitosis is an equational division. On the other hand, if an organism to be formed is a haploid organism, then the zygotic cell would undergo meiosis, because meiotic division is a reductional division. So whether the zygote forms the embryo through meiosis or through mitosis, both of these divisions results in an increase in cell number. But after a certain period of time, the embryonic cells starts to mature and specialize into tissues and organs. And this process of maturation and specialization is called as cell differentiation. Now, as shown here, Some of the embryonic cells can give rise to different types of tissues or organs, such as heart, nerve cells, or blood cells, or skin cells, muscle cells, etc. On the basis of the site where embryo development takes place, animals can be classified into oviparous, viviparous, or ovoviviparous type. Oviparous animals lays their eggs and their embryo development occurs outside the organism in their hard-shelled eggs. In viviparous animals, they give birth to young ones and the embryo development occurs inside the female body. Interestingly, there's another group of animals called as ovoviviparous. The eggs in these animals are retained in their oviduct and the embryo develops in the egg. The eggs, on hatching inside the body, would gradually develop and later these animals will be giving birth to live young ones. And such phenomenon is seen in cases of some sharks, some snakes, as well as in some other animals. So in the flowering plants, after fertilization, several changes occurs in the flower. The outer three whorls of a flower, the whorls of the sepals, petals, and the stamens, eventually starts withering and falls off. The pistil then undergoes modifications, including the matured ovule becoming into the seed and the ripened ovary changing into fruit. And the fruit gets surrounded by pericarp, which is formed from the ovary wall. The fruits are advantageous to the plant as it helps in better seed dispersal and thereby results in the generation of new plants. Now here's a figure taken from your textbook of some fruits with a label S and P. S is for seed and P is for pericarp. Now this figure should remind you of our last year lesson on placentation. We learned that placentation is the attachment of an ovule or a seed to the walls of the ovary. And we also learned about the different types of placentation. Now you will study the figure carefully and identify the types of placentation in them. Well, with this, we come to the end of my presentation on fertilization and post-fertilization events. And please feel free to ask questions on this presentation on our WhatsApp group. And note the assignments and the due dates here. And I hope to see your work well on time. So till then, stay safe